And now we're going on to the first session discussing in more details about autism and CFTR. And I would like to invite our first speaker, Dr. Ilan Dienstein from Gurion University, who's going to tell us about from sleep recording to motion capture, developing objective quantitative measurement for symptom severity in children with autism.
Thanks. Hi, Deepa. So it's a pleasure to be here. I'd like to tell you a bit about phenotyping autism and do it in the context of this autism center that we've been building in the Negev over the last several years. The Negev is the southern half of Israel, for those who don't know. So I think that autism, unlike other perhaps neurodevelopmental disorders, there's this growing consensus that autism is very heterogeneous both in the mechanisms that create autism and in the phenotypes. There are many ways of being autistic. So you can see, and I'm basing this claim on the genetics from the last several years, and most of you might be familiar with syndromic forms of autism like Rett and Fragile X, and this might not be the PMS you're thinking of. This is Phelan-McDermott syndrome. So these are specific mutations in specific genes, and there the mechanism is tractable, and you can think about therapeutics. But in the vast majority of other autism cases, right, we don't know much. And in order to make headway and identify specific forms of autism, it would be extremely important to characterize this heterogeneity in a wide variety of fields, ranging from behavior to physiology to genetics. And some of these measures, you know, are more objective. I just wanted to point out that in behavior we have this added complexity that the measures are mostly subjective. They depend on parental reports and clinical evaluations. And so part of what some of the examples that I'll show you in this very fast 15-minute presentation have to do with how to quantify behavior in a more objective manner and also longitudinal manner that might be useful for characterizing this heterogeneity. And just to point out that the main goal here is to relate things. So you would want to know how children with certain genetics and certain physiological measures, what kind of behavior do they manifest and whether they're distinct, okay, and likely to benefit from different treatments. And in order to do that, it's really important to collect all of these measures from the same individuals. You can't have one lab working on genetics and another lab working on physiology on separate cohorts, because you won't be able to relate the data. And not only on the same individuals, but you would want to also capture this data in a longitudinal fashion, because symptoms change, behavior, physiology, everything changes throughout development, particularly early in development, which is perhaps the more beneficial focus for therapy. So this was very strong motivation to build an interdisciplinary center at the university, this was kind of a grassroots effort that started 5 years ago. I just wanted to point out two important partners in this, who are Idan Menashe, who's a geneticist and an epidemiologist, and Gal Meiri, who is the child psychiatrist in Soroka. And there's a list of about 20 other partners. And so this is the unique thing here is the interaction between the physicians from Soroka Medical Center and the researchers from Ben Gurion. And there are a very wide variety of fields, including things like biomedical engineering and neuroscience and so forth. And these are exciting times for us because we just received this national grant from the Ministry of Science, which is important because this is, I think, the first time that the Israeli government actually put money into infrastructure for autism research in the country. And so the idea is to turn this center in the Negev into a national, to have other sites and turn this into a national center. And I think the reason they selected us was that there's this close proximity between the hospital and the university. So one of the reasons why they chose is that this makes the research a lot easier. And I want to tell you a bit about how we do things. So Soroka is where we operate. We have rooms inside the hospital. It services, it's like a healthcare monopoly in the Negev. It's the only place where you can get major medical treatment. Services about 1 million people. And there's somewhere between 180 and 200 new cases of autism there every year that are diagnosed. We've incorporated, for the last 3 years, we've had research assistants and clinicians from the center who are inside the clinics that perform the autism diagnosis, and this relationship is very important for us because that's how we recruit the families. We have about a 75 to 80% recruitment rate, which is really important. We make the research very easy for the families. We do all the research in rooms that are adjacent to the clinics. So we have about 14 rooms in the hospital. We've turned one of them into an eye tracking lab. We do motion capture in the ADOS room that I'll show you in a second. Looks something like that. We do work in the EEG. We record EEG in the sleep lab in Soroka. This is my 70-year-old who I've convinced that going to the sleep lab is the coolest thing in the world, very useful. And we also do genetics in the sense that we collect saliva samples, we extract DNA. We're part of the Autism Sequencing Consortium, which is a US-based effort to characterize 50,000 individuals with autism. And now we just got clearance to join Midgam, which some of you are familiar with maybe, which is the National Biobank. And so we're about to start collecting blood and urine and things like that. Okay, this is our workflow. Just I wanted to mention here that except for the initial diagnosis, we try and capture the families as early as possible. It involves several meetings, which gives us lots of opportunities to recruit them to the research efforts. The families come back every year, and we've just incorporated follow-ups that include ADOS and a variety of standardized assessments every year, so we get this longitudinal measures, and we're really hoping to kind of copy this workflow and do the same thing in Asaf HaRofeh Uditza soon, and so there will be additional sites in Israel over the next 3 years. This is our recruitment rate so far, so we have over 600 families in this. This is the age where we typically recruit the children, so you can see a nice early age distribution in the Negev. A quarter of these kids are Bedouin, who are very unique in that 2/3 of the parents are first cousins. So this is a unique environment to do genetics research in. And a huge advantage is that the majority were born in Soroka, so there's access to all the maternity ward, neonatal care units, so lots of details about potential environmental risk factors, and if you're thinking about specific mechanisms in autism and how to relate them to later development, this is very unique information. There are even ultrasound scans of the baby from high-risk pregnancies. It turns out that about 20% of our cohort are high-risk pregnancies, and there's a similar percentage of kids who were scanned with MRI during the first year of life for clinical reasons, right? Nothing to do with us, but really unique information to connect with the later phenotype. Of the different types of data that we collect, this is what we have so far, so we don't have all of the data types on all of the children. We have about 180 trios, so child and both parents who gave us saliva samples for DNA. I won't have any time to show you eye tracking results. This is a really interesting direction of research for phenotyping the children early on. I'll show you a bit of motion tracking. We do speech recordings. We analyze the speech and see what the child says. We identify things like echolalia, which is a form of repetitive behavior. Ilan does a lot of work on the clinical records. I'll show you a tiny bit of EEG recordings in sleep. We haven't looked at any of this yet. Lots of questionnaires, and the biobank is coming soon. This is the follow-up data, so we already have some interesting follow-up information. We're using this to determine how symptoms co-develop throughout life. So if there are changes in sensory sensitivities and cognitive abilities, how does that relate to the autism severity, which is measured with the ADOS, and you can also break that down into different subparts. Okay, the idea is to turn this database into a national database and make it freely available, at least some parts of it, to all the researchers in Israel. So I want to show you a brief example of how we do motion tracking, which is one, I think, form of deep phenotyping, which I specifically hope will be useful. So what we've done is, I need to tell you that we have this is the ADOS room where we do the assessments. We have, in addition to HD video cameras and microphones and a bunch of other things, we have Kinect cameras around the room. So we have 4 synchronized Kinect cameras that give us depth information, which is very useful for identifying individuals in the room. And it looks something like this. Shouldn't have done that. Okay, so what you'll see on the right in a second is this is the student, Ilan, who was very happy with himself, and a great student. So this is the view from a single camera. This is after incorporating all 4 cameras. You can identify individuals. You can fit kind of an avatar or skeleton to both of them. This is the child. This is an older child, 6 or 7 years old, and that's the clinician. And after identifying the individuals, you can start quantifying specific measures, such as how much the child approaches or avoids the clinician. So you see here kind of a dynamic measure of whether the child is walking towards the clinician or away from them. You can measure the distance between them. Okay, and we do all of this only in the segment of the ADOS where the child is freely playing, which is about 5 to 10 minutes, depending on the individual. And then a third interesting measure is whether the child is facing the clinician, so we can compute a vector of where the child is facing and see how that interacts with the location of the clinician. See that here. Okay, it looks something like that. And then the bottom line is that combining all of these measures together explains about 30% of the variance in the ADOS scores. So there are different relationships here, but I just wanted to point out that we're thinking of this as an initial kind of proof of concept that using this kind of technology, and by the way, you don't necessarily need fancy Kinect cameras. You could potentially do this with a bracelet on the clinician and the child. That might be enough. You could also then think about turning this into something you would put in a kindergarten and have bracelets on the kids in the kindergarten and see how they move in space and avoid or approach each other. So these are kind of the dreams we have about this. I don't know how much time I have left, but 3 minutes. Okay, so I'll tell you very briefly about sleep, which is another area that, so thinking about this deep phenotyping, right, and measures that are, another nice thing about the Kinect is you can have the child come back and you can measure this longitudinally and see if it changes and whether these changes correspond to other changes in the child's symptoms. Okay. So that might be really useful for clinical studies as well. Another measure that's perhaps even more useful is measuring sleep and seeing what happens. So there are huge sleep problems in autism, particularly early on. This is a parental report from the kids in our database. You can see that about half of them have these very high scores in sleep disturbance, okay, in comparison to a typically developing group that we collected a while back. Despite this, only a small minority are actually treated, mostly with melatonin, which is a mild form of medication. There are very few other options. Perhaps medicinal cannabis might be one. We've shown that there's a relationship between the amount of sleep disturbance and sensory sensitivities, in particular to touch. So there's a strong correlation. You see these kids with autism who have very severe sensitivities to touch. They tend to have the severe sleeping problems, so that might be a subgroup of individuals with autism who would benefit greatly from sleep or sensory therapies, rather than, for example, testing everyone, which is the way most clinical studies work today. And then a subset of these 350 kids who filled out the questionnaires, about 40 of them participated in an overnight EEG. In general, I'll tell you that we were surprised at how successful it was to complete. 38 of the 40 completed an overnight sleep exam. They slept a bit less, about 40 minutes less. They tended to fall asleep later. And to make a long story short, the main finding is that they have less slow wave activity, which are these initial early stages of sleep in the beginning of the night that tend to be very deep. And so when we look at the both at the amount of segments that were scored as deep sleep, there's less of them in the kids with autism. If we quantify the EEG oscillations in these very slow frequencies, which are apparently important for lots of things, there's less of them in the first half of the night. And when we look at individuals, there's this paradoxical issue that, so the deal with slow wave sleep is that if you look in the literature, this is a person sleeping during the night, the slow wave activity drops to 20% of what it was when they fell asleep, and then during the day it increases. It's a form of sleep pressure. So if you're sleep deprived for 24 hours, you'll have slow wave activity which is much higher. In the kids with autism, those who fall asleep later, they should have larger slow wave activity because they're not sleeping enough, but they have smaller oscillations, weaker oscillations. So this suggests that the mechanisms for driving sleep pressure and creating the slow wave activity is not working there properly, and that this might be a way of, and by the way, it's not only correlated with their sleep in the lab, that's it? Okay. So I just want to say that I think sleep is an incredibly important topic that's underexplored in autism, and it might actually be one of the sources of the behavioral problems in some of the children, not all of them, and that's an agenda that I really want to pursue at the center. So incorporating all these measures, thinking about how to use them for measuring clinical success in subpopulations of kids with specific symptoms, specific genetics, and so forth. That's the direction this is going to. And thank you very much. Timid crowd, I see.
I have a question.
Sure.
So, did you look at fine motor skills in young children and how that correlates with the severity?
So we initially wanted to use the Kinect cameras to actually look at kinematics of movements. I come from a motor control background also, but the Kinects they have very poor accuracy in that they sample kind of roughly. So it's easy to get these rough measures of where the child is in the room, and it's a lot more difficult to see more precise movements. And we haven't been doing any of the behavioral fine motor control. I mean, it's part of some of the standardized tests, but we haven't been developing that. But that's interesting, yeah.
So your data on sleep is really exciting. So it could suggest maybe that this kid has really a problem with sleep homeostasis. So my question, I don't know if you can get approval for this, but if you, for example, can do sleep deprivation in these kids and monitor simultaneously EEG activity, do you see abnormal activity during sleep deprivation? So I would guess.
So I was
I don't know how EEG will be
something simpler to be. So one of the directions that we're pushing is using a bunch of, there are all these gadgets now with these new technology to measure sleep at home over time. You could potentially use that to measure temperature cycles over the day and see how their circadian rhythm is. So there's controversy over whether the problem is sleep pressure or circadian rhythm dysregulation. Those are two options for what might be driving these results. And you could get at that with some measures at home. Doing a sleep deprivation study would, of course, be much more difficult in these ages.
I just doubt if you can really detect it with these tools, because, for example, in Alzheimer's model, what we do, we see really very local changes inside of the brain during all of this activity. So it's more difficult. Thank you very much.
Thanks. Okay.
Thank you very much for this very important topic by all, point of view. But I just wanted to ask a practical question. You were talking about the cohort that you are trying to put available. How, I'm not speaking about the imaging, I'm speaking about the other clinical and other data. How do you keep it so we can use it from any place? How do you?
Well, they're very. We're not the first to do this. I mean,
I know, but there is a big problem. Yeah.
Not really. I think that the Simons Foundation have kind of broken through with this. There are now cohorts of thousands of kids who are easily available to any researcher in the world. It's just an issue of getting the IRB and Helsinki protocols to a point where the families agree to it. It's all anonymized
okay,
data.
so they are signing, each one is signing?
Yes, of course. When we recruit a family, they sign.
I wasn't sure. You were talking about the clinics, therefore I asked.
No, all of this. So 80% of the clinic is research data where the parents have signed off and given us clearance to use this. Okay.
Thank you very much.